Let's start by understanding what sterilization really means. It's the process that completely removes all microorganisms from a surface or substance. This isn't just about cleaning. Sterilization ensures that even the toughest forms, like spores, which can survive harsh conditions, are eliminated. These spores come from various organisms, like bacteria and fungi, and are designed to survive until conditions improve. So, sterilization is crucial to stop any chance of these microbes coming back. Sterilization means making something completely free of all microorganisms, including spores. Now, spores are special because they're designed to survive tough environments and can spread easily. They're part of the life cycle for many bacteria, plants, fungi, and even some protozoa. Understanding this helps us see why sterilization has to be thorough, targeting not just active microbes, but these resilient spores too. Now let's talk about how sterilization fits into the bigger picture of controlling microbes. There's a hierarchy, starting with sterilization at the top, which completely eliminates all microorganisms. Below that, we have disinfection, which removes pathogens but not all microbes. Then comes decontamination, antisepsis, and finally cleaning, which mainly removes visible dirt. Each step has a different level of control, and sterilization is the most comprehensive method we have. To control microbes, we use various antimicrobial agents, each with a specific role. Some kill organisms outright, like biocides including chlorine. Others, like bacteriostatic agents, prevent bacteria from multiplying. There are narrow-spectrum antibiotics targeting specific bacteria, and broad-spectrum ones that cover many types. We also have disinfectants for surfaces and antiseptics for tissues. Understanding these categories helps us choose the right agent for the job. Physical methods offer diverse ways to sterilize materials. Sunlight, especially UV rays, can reduce microbes. Heat, both dry and moist, kills microorganisms through protein damage. Radiation uses energy waves to sterilize, while drying removes moisture needed for growth. Filtration physically removes microbes, and vibration uses sound waves to disrupt them. Each method works differently, offering options depending on the material and situation. Chemical sterilants work by targeting crucial components and processes within microorganisms, leading to their inactivation or destruction. Here are the key modes of action. Denaturation of proteins. Chemical sterilants, like heat, disrupt the 3D structure of proteins, causing them to lose their function. Damage to nucleic acids, DNA or RNA. Certain sterilants, like ethylene oxide, can alkylate DNA, proteins, and other cellular components. Disruption of cell membranes. Sterilants such as hydrogen peroxide and ozone can damage cell membranes, leading to loss of cellular integrity. Inhibition of essential metabolic processes. Some chemical sterilants interfere with crucial biochemical pathways within the microorganisms, preventing them from synthesizing essential components or generating energy. Let's dive deeper into some physical sterilization methods. Sunlight's UV rays damage microbes, but effectiveness depends on location and exposure time. Drying inhibits bacteria since moisture is essential for their growth, though spores can survive this. Heating, whether dry or moist, kills microbes by denaturing proteins and causing oxidative damage. The temperature, exposure time, and the type of material all influence how well these methods work. The red-hot method is a straightforward sterilization technique. It involves heating an object in a Bunsen flame until it glows red along its entire length. This is commonly used for metal tools like inoculation loops, forceps points, and needles. The intense heat effectively kills all microbes on these instruments, making them safe to use in sterile environments. Flaming is another quick sterilization method. Instead of heating an object until it's red hot, you expose it to a flame for just a few seconds. This works well for items like glass slides or the mouths of culture tubes. It's a practical way to reduce contamination quickly without damaging the material. Incineration takes sterilization to the extreme by completely destroying material through burning. This method reduces waste to ashes, making it ideal for disposing of pathological wastes, surgical dressings, and animal carcasses. The high temperatures ensure that all microorganisms are eliminated, preventing any risk of contamination from these materials. Hot air ovens sterilize by circulating dry heat evenly inside a chamber. Fans help distribute the heat, and a thermostat keeps the temperature steady. Different temperature and time combinations are used, like 160 degrees Celsius for 2 hours or 180 degrees Celsius for 30 minutes. Controls include biological and chemical indicators to confirm sterilization. The key concept here is thermal death time, which is the minimum time needed to kill microbes at a set temperature. When using a hot air oven, certain precautions are important. Avoid overloading the oven because it can block heat circulation and reduce effectiveness. Material should be completely dry before sterilization, as moisture can interfere. Also, avoid rubber and plastic items since they can be damaged by the high temperatures. Following these guidelines ensures the sterilization process works as intended. Moist heat sterilization methods are categorized based on the temperature used. Below 100 degrees Celsius includes procedures like pasteurization, which is commonly used for milk and other liquids. Moist heat at 60 degrees Celsius or vaccine bath can kill most mesophilic non-sporing bacteria with an hour exposure. Certain media can be sterilized using a process called inspissation, which is an example of fractional sterilization. At 100 degrees Celsius, boiling is a common method, with tyndenzylation being another method of fractional. Above 100 degrees Celsius, methods mainly include autoclaving, which is a steam under pressure method. Autoclaving is considered the most effective moist heat sterilization method, as it utilizes saturated steam under pressure to achieve temperatures above the boiling point of water. An autoclave, similar to a pressure cooker, creates a high-pressure environment that allows steam to reach temperatures of 121 degrees Celsius or 134 degrees Celsius. Saturated steam efficiently transfers heat, leading to rapid penetration and killing of both vegetative cells and spores. Filtration and radiation are advanced physical sterilization methods. Filtration uses various materials like earthenware, asbestos, and membrane filters to physically remove microbes from liquids or air. Radiation comes in two types: non-ionizing like UV rays that interfere with DNA replication, and ionizing, such as X-rays and gamma rays that destroy DNA directly. These methods are especially useful for sterilizing heat-sensitive materials and environments. A Seitz filter is a type of depth filter historically known for its use in sterilizing heat-sensitive liquids. A HEPA filter, or high-efficiency particulate air filter, is a specialized mechanical air filter widely recognized for its ability to remove microscopic particles from the air. Chemical sterilization involves several types of agents. Heavy metals have antimicrobial properties, while oxidizing agents include halogens and hydrogen peroxide. Alcohols like ethanol and isopropyl are commonly used. Alkylating agents such as formalin and glutaraldehyde modify microbial DNA. Gaseous agents like ethylene oxide sterilize by penetrating materials. Phenols and phenolics act as bactericidal agents. Together, these categories cover a wide range of chemical sterilizers.